Good afternoon and welcome to Isopogen, which is a privately owned company developing life-changing cellular medicines. The company's patented StemSmart technology improves the clinical efficacy of mesenchymal stem cells, offering a powerful competitive advantage against other MSC products. The company's proprietary cellular medicines made using the StemSmart technology are in late-stage clinical development to treat several autoimmune conditions, including Crohn's disease, a debilitating gut illness with a global market worth $7.5 billion each year. Already more than 150 patients have been safely treated with the products created using the StemSmart technology in clinical trials and under special access arrangements. These results have been widely published, including an impressive 80% clinical response and 53% clinical remission in life-threatening or refractory Crohn's patients in a phase 2 study, making the StemSmart created product the only MSC treatment clinically proven to treat this condition. Today from the company, we have Dr. Marion Sturm, the CEO and Chief Scientific Officer, as well as the CFO, Paul Fry. Marion is an expert in research, operational and clinical development of cellular treatments. She is a facility director of Royal Perth Hospital's Cell and Tissue Therapies in WA, and she's also a member of the TGA Advisory Committee on Biologicals and Senior Research Fellow with the Centre for Cell Therapy and Regenerative Medicine at the School of Biomedical Sciences in UAA. Just in terms of running today's session, I'll hand it over shortly to Marion, but if anyone has any questions as we go through, please do so via the Q&A box. But Marion, I'll hand it over to you to get started. Thanks very much.
Thank you. It's a pleasure to be able to present to you all today. So I'll just start and go through the disclaimer and then tell you that Isopogen is about life-changing cellular medicine. It's actually called a mesenchymal stromal cell, or we shorten it to MSCs. And our company is underpinned by the StemSmart technology. And for those of you that don't know, I'm sure most of you don't know about mesenchymal stromal cells. They are remarkable and powerful cells. They have attracted significant clinical interest. These cells are adult stem cells that you can get from lots of different tissue, but most work's been done on bone marrow. As I said, they're a stem cell. They can divide and they can turn in other cell types. And in the lab, we turn them into fat, bone and cartilage, just as part of the characterization. They offer two primary clinical properties, and that is that they're involved in tissue regeneration. They repair damaged tissue, and they've got immunomodulatory, anti-inflammatory capabilities, and they do this by interacting with most of the cells of the immune system to bring it back under control, and by releasing compounds and factors that mop up inflammation. And those two properties are intertwined because inflammation occurs with all injury. The other remarkable property of these cells is that they are what they call a universal donor cell. You can put anyone into anybody else's. That means there's no need for matching. Our mesenchymal StemSmart product is obtained from bone marrow and manufactured according to our patented process to produce a more efficacious cell with the potential to treat a variety of immune inflammatory disorders. And there's plenty of possible clinical applications for the cell. There's been a significant focus on these cells worldwide and a lot of trials with a lot of promise. However, there's been few products that have actually been approved. Commercialisation has been slow coming, and there's several reasons for this. Predominantly, all the work in MSCs has been institutional-based trials, early trials with limited funding. The cells are quite expensive, and there's been inconsistent clinical outcomes over the various studies. As I said, most of them are early stage and underfunded, and there's been a diversity of different types of MSC products. I said that you can get them from lots of different tissues, and the different tissue types have slightly different properties. And of course, they've been used over an array of clinical conditions. So what I'm trying to tell you is there hasn't been a lot of consistency. There's the few phase 3 trials conducted have actually not been that successful. So that's actually detracted from the full commercialisation of the cells. There's certainly manufacturing challenges when you make a cell therapy. You've got to have access to certified manufacturing facilities and be compliant with good manufacturing protocols. And there have been various types of protocols used to manufacture the cells, and most of the manufacture has been unregulated by authorities, and upscaling to produce large amounts of cells has got its own challenges. In about how do we come to develop the technology? Well, started in, I sort of worked in hematology, and it started in 2004 when a Swedish physician demonstrated that if you put these cells into patients with a refractory graft versus host disease, which is a complication of having a bone marrow transplant, they could actually bring the GVHD under control. And this was a pivotal case. Hematologists came back from a meeting and they said, you know, this would change how we treated GVHD. And so in 2005, I began the development work of MSCs at Royal Perth. And in 2007, we treated our first patient. GVHD is when the new blood system rejects the recipient, and it affects the skin, the gut, and the liver predominantly. And for those patients who fail treatment, it's terrible for the families, the staff, and the patient themselves. So there's no other treatments available. So we treated the first patient, a nice young man, and he was in ICU with a very grim prognosis. And he had a number of treatments, and he recovered, and he was discharged to the ward, and he eventually discharged from the hospital and is surviving to this day. And that led to a clinical trial in what they call refractory non-responding GVHD, and it extended to children as well as adults. And the success of that trial and the safety profile, it encouraged other studies and other disorders, because a lot of the features of GVHD you see in other conditions. For example, patients sometimes get a lung condition for fibrosis of the lung in GVHD, and so we started to do a trial in lung rejection, which you also get fibrosis. They get gastrointestinal problems, which is like you see in Crohn's disease. So we did a trial in Crohn's disease and various others. So we've actually done 11 clinical trial studies, treated well over 170 patients and given them more than 1,100 infusions. It's been a patient-driven technology, so there's a need to do it. It's been done in the public health system, and it's really demonstrated safety and efficacy, and it's addressed acute clinical needs. We have sort of technology around the development of this cell, and so in 2014 I filed a patent over the manufacture, which was transferred to the company in 2015. And so to give you a bit of an idea how we make these cells, it's sort of fairly standard process where you obtain some marrow, you isolate the cells, and then you basically grow them up in number. And then the magic comes in at the engineering step, where we incubate the cells in a special proprietary media that activates the cells, and it does something else, which I'll tell you about. And then we go on and after they've activated, and collect them, and we freeze them and store them, and then they're then available to administer to patients. So why have we done so well? Why do our cells work perhaps better than lots of the other people that are working in this field. Well, one of the things is that we've always had a strong manufacturing process. That means we've got good quality products. They're manufactured to strict protocols. They're made in a very accredited laboratory. They've had extensive testing. They're not extensively expanded or multiplied. They don't go out too far. From 10 mils of donor marrow, we can make well over 200 doses, and they're delivered via very simple intravenous infusion that's quite safe. The patented technology includes the removal of bovine proteins. Pretty much everybody in the cell therapy area grows their cells in fetal, what they call fetal calf, but it's bovine proteins. They stimulate growth, and about 50% of people actually have antibodies to bovine proteins. So you've got to take them out of the final cell preparation, otherwise the cells are going to be automatically rejected by the body. So most people just wash the cells and wash the bovine proteins off, but there's a lot of the bovine proteins that are within the cell. So what we do is we actually metabolize any remaining proteins out. And so the levels of the bovine proteins is minimal. And the bit of engineering we do in that proprietary media makes the cells primed and ready to respond to inflammation. And we've done various testing in the lab that supports that. When you put the cells into people, they're anti-inflammatory immunomodulatory actions triggered by the exposure to inflammatory molecules. And so, they have to basically be switched on. So ours are ready to be switched on, and they are 3 times more potent than cells made in conventional methods. And as a result of this, we've had standout results in clinical trials, particularly in Crohn's disease. So we've got a strong intellectual patent profile that covers key jurisdictions with patents that go out to 2034. And StemSmart's been used in a variety of indications, and these include COPD, chronic obstructive pulmonary disease, which is emphysema, a tissue transplant rejection, which is lung transplant, and also acute kidney rejection, inflammatory bowel disease, it's Crohn's disease, and in, I said, graft versus host disease, it's when you're a complication of having a bone marrow transplant. The product is an allogeneic product, which means it comes from somebody else, and there's no need for matching, so it's actually available off the shelf or out of the freezer in our case. As I said, we've used it in 11 clinical trials or studies undertaken in Australia with a lot of patients safely treated. It's clinically shown to treat a variety of disorders. And we would like to initiate some, further phase trials in particularly a phase 3 in non-refractory Crohn's disease. Up to date, we've basically had the Crohn's patients who have failed every other treatment, so their only option is therapy. So we want to actually come in a little bit earlier in their disease condition and hopefully save them from some of the complications of the more advanced therapies. The other indication for which the cells are likely to have a significant effect is in COVID-19. And we would like, well, if we had any patients, we would really, we have initiated a trial in COVID-19, but we're waiting on recruitment. So these basically are a list of the clinical trials and studies that we've conducted. You'll note that they're mostly phase 1 and phase 2, most in immune inflammatory disorders, apart from one which is in tissue regeneration, which is cranial reconstruction, making pieces of bone. And then next year we're seeking TGA approval for refractory Crohn's disease, graft versus host disease, and kidney rejection. And this just shows you the conditions we're addressing, and they all have unmet clinical need and significant markets. And for those that don't know Crohn's disease, it's a terrible disorder. It's a chronic inflammation of the gut. It's a lifelong condition, and eventually 30% will require surgery. And we've had excellent clinical results with our cell therapy. And globally, Crohn's is a significant problem with 2 million people worldwide. It's a growing market of westernisation, and you can see that we have patent coverage in key jurisdictions. In terms of Crohn's disease, you start with antibiotics and anti-inflammatory agents and immunomodulators, and then what they call the biologics, which is a antibody to one of the inflammatory markers, was a big game changer for the treatment. And those biologics represent 60% of the market, but 30% of patients will lose response to them and go to theatre. And so we're looking to come up against the biologics. I think I'll skip, in the interest of time, talking about kidney and rejection and GVHD. But as I said, we're looking to gain license to market in 2021. There's a number of lung disorders that have or are in progress. And the current one is a phase 2 in chronic lung allograft dysfunction. And so far we have recruited, I think, 44 of 82 patients. It's an Australian-wide trial, so and it's a placebo-controlled, so it's a really good trial. And in terms of moving forward, we have the ability to generate revenue by a variety of means, and this includes manufacturing, distribution, and marketing sales, licensing out our technology or doing tech transfers, and also by licensing out our patents. Got a board of management. Myself, I'm an expert in cell therapies. Paul Fry, who's our secretary and chief financial officer, Rebecca McCracken, and Rob McKenzie, who are both lawyers, and Rebecca having had significant patenting experience, and Rob a very senior lawyer. Currently we have 113 million shares on offer and net cash of 1.4 million. And so why should you invest in Isopogen? We've got a global patent portfolio around our manufacturing process. It's shown to improve the efficacy of mesenchymal stromal cells. We've got an advanced product portfolio covering multiple indications. We've treated quite a lot of patients with various ailments. And so we've basically got a phase 3 ready biotech that's seeking TGA approval for those critical conditions where there's no other therapies available. There's large addressable global markets for multiple indications. We've got a good safety profile. We've got existing license agreements in place with Royal Perth Hospital, a TGA licensed manufacturing facility, and we've got management and board capability. The key risks are listed there. So thank you. I'm happy to take any questions.
Well done, Marion. Very well done for the first time virtually, particularly in what is a relatively complex. You made it easy to understand. So well done. Just in terms of the company's funding history, perhaps you could just give. I know that the company can't make any comment on any sort of potential IPO as it's assessing different capital raising opportunities at the moment. But can you just give us a bit of a background in terms of the funding profile of the company, please?
I might hand that over to Paul, my Chief Financial Officer.
No, in terms of the funding, the most relevant is really the recent round. We did a round with Bell Potter. We raised basically that $1.4 million, and that was priced at 10 cents, which gave a valuation of the company of $10 million. So we've got 113 million shares out there. It's very tightly held. and the recent financing, we issued 13 million shares. So prior to that, there was 100 million. Now we've got 113 in place.
Right, thanks.
So those funds are going to be used to progress. We are going to pursue a listing on the stock exchange, progress towards IPO, and progress the commercialisation of our technology.
Great, thanks, Paul. And just in terms of what are the major advantages in using your manufacturing process versus your peers?
Well, ours has had, we've got consistent clinical results, but more that our cells are more potent, I think, than any of the others out there. And that's because of the magic around the manufacture, what the patent covers.
Right, and in terms of, I know that you've just raised, roughly that $1.4 million, but any subsequent larger capital raising, whether it was part of an IPO process or otherwise, what would that sort of funding be used for?
So in terms of that, the next funding raise will be through an IPO, and that will be to do a number of things. One is to progress the a phase 3 trial or phase 2-3 trial in Crohn's disease. So at the moment we're applying for refractory Crohn's, which we have the funding in place to do that. But what we'd like to do is do a fairly big trial in Crohn's to go up against the biologics. So that would be a phase 2-3 trial, and that'd be significant cost. And then the other thing in the immediate term is to then also look at doing a fistulas trial in Crohn's. So those two are key. And then the third thing is, if we get up with the TGA, which we're hopeful we will do, then we'll take that to the US hopefully, and then try and treat refractory Crohn's in the US. So that's the regulatory commercial path as we sit here.
And if you don't know what fistulizing Crohn's is, it's a complication whereby you basically get a hole in your gut, and your gut discharges externally through the skin or into your abdomen or other areas. It's quite difficult to treat and a significant problem.
Right. Next, another question was when will you IPO?
Well, probably in the next 6 months, the first 6 months of next year. That's the plan at this stage.
Okay. Another question: are you doing any collaborative work with a chap by the name of Michael Winlow or Emerald Clinics?
No, not at this stage.
No worries. If there's not any other questions that come through in the next little while, we might finish up there, and I'll pass back to you to summarise. But it was certainly very insightful presentation, and one that I personally probably have to go back and listen to again in terms of learning. So I'll hand it back to you to summarise, and we'll finish up there. Thanks very much for your time.
Thank you. So I hope I've shown you that I've shown you a large amount of work that's gone on for well over a decade, that a technology around a cell therapy that significantly changes the lives of patients, for which there are no other alternatives. So I think we've worked hard in developing the company and translating the technology, so I can see a great opportunities for us.
Great. Thanks so much, Marion, and thanks so much, Paul. And if anyone wants further information regarding staying in touch with any future funding plans, please feel free to reach out to Marion and Paul with their contact details there, or if you've got my email, simon@nwrcommunications.com.au. Thanks very much, guys, and enjoy the rest of the afternoon.
Thank you.
Bye-bye.